Hey everyone, so this video we're going to look at the polymerase chain reaction. It's often going to be referred to as PCR, and when I talk about PCR in this video, I'm going to refer to that. But it's a part of our biotechnology unit that we're going to look at on a way that humans are starting to manipulate DNA. So our intentions for this video is to understand the purpose of why we use a PCR reaction and to then know how that reaction actually occurs. So we'll start off with the human genome. Obviously, we've got a large amount of DNA and a large amount of genes. Now, often when scientists are working with DNA, they might only need a very, very small section to work with. So we use the PCR reaction to actually amplify that DNA to a level that it can actually be worked with a scientist. So having just one copy of a gene is so small that we couldn't actually do anything with it. So we need to amplify that to make many copies of the same section. It could also be the scientist has maybe, for example, a forensic scientist has found a very small section of DNA at a crime scene. They would need to amplify that section to be able to actually work with it and test it. So the PCR reaction is just an amplification process. So for PCR, we need a couple of things. We need our original DNA sample. We need these molecules known as DNA primers. We need nucleotide bases, so our A, Cs, Gs, and Ts. And we need a DNA polymerase, and often you might see it referred to as Taq polymerase. That's just a type of DNA polymerase we're going to be using. And lastly, we need heat. So those molecules we're going to go through and discuss how it actually works to make the PCR reaction. So there are four main stages. We've got denaturation, which is where DNA strands will actually separate. We've got stage 2, which is annealing, which is where those primers we talked about start to work. And then we've got extension, which is where our Taq polymerase or DNA polymerase starts to copy our DNA strands. And we repeat that process over and over. Now you'll notice that in our steps, I've written down approximately 90 degrees, approximately 50, and approximately 72. That's because they're not necessarily set temperatures. They're around that temperature. And depending on which textbook you read or which exam question you come across, they sort of have different temperatures for those stages. Now, the numbers 90, 50, and 72, they're from the VCAA, examiner's report. So we're going to use those numbers as our approximate numbers. So you can see in our diagram, we've got denaturation, annealing, and extending. Now the stage that isn't shown is stage 4, which is just a repeating process. We repeat it over and over again. So we'll get stuck into the different steps. So the basic process is that we have our DNA sample. Now this might be a sample that we've taken from, let's say, a crime scene, or it could have been a certain part of DNA that we've cut out using restriction enzymes, okay? So the first stage is our denaturation. So we're going to heat the DNA sample to 90 degrees, and that's going to break the hydrogen bonds that are between the complementary base pairs. And when we heat those bonds, we end up separating the double-stranded DNA into 2 single-stranded pieces of DNA. Our next step is the annealing stages, where our primers will join DNA at opposite ends, and again it occurs at around 50 degrees. Now these primers attach on either side of the section that we want to replicate. So there's all different types of primers. In this example, the primer might be a set of bases that are reading as TTT, and they're attaching to either end of our sample we want to amplify. And then lastly, our last stage is our extension stage. So our Taq polymerase will bind to our primers, and as you can see, it's then going to start reading along the bases. And much like RNA polymerase does, it's going to start adding complementary bases. So you can see here now, we've now got, okay, complementary change. And this process occurred at about 72 degrees as well. So you can see that we now have got double the amount of DNA sample that we originally started with. Okay, so there, just in a visual, we can see we started with 1 sample, we've now got 2, and we then repeat this process, that denaturation, the annealing, and the extension over and over and over again until we get a large amount of copies. So here you can see, for example, we've been through the process of denaturation, annealing, and extension or elongation, and then we're going to repeat that process over and over and over again. Now, obviously, if we start counting, we're going to start with 2 on our first cycle, then 4, then 8, and so on and so forth. So eventually we end up with millions of copies after not very many cycles of PCR. We also use a machine called a thermocycler. Now this is a machine that will cycle between the temperatures that we need for the PCR reaction. So it will go from 90 down to 50 up to 72 and just keep repeating that process over and over. And that allows the stages of PCR to occur over and over again. And that's going to be a really important piece of machinery that we're going to use to conduct the PCR process. Now, some of the positives of PCR is that we can have a very small sample of DNA that can be amplified very, very quickly and very efficiently. So it doesn't take a large amount of vigorous work. It's just a process of adding those ingredients, adding the DNA sample that we want, and letting the process occur over and over and over again. Some problems with the PCR is that we can actually contaminate the DNA sample that we're amplifying. So if the scientist isn't careful, their own DNA can be amplified and then placed into the final product. So obviously they're going to be very careful with how they complete the process. And the other negative is that we can't actually test to see if it works until after the process has already occurred. So you might have wasted your time doing the PCR process to not even realize that some of your ingredients weren't correct, or maybe you had the incorrect primers, and you wouldn't know that until after the actual process has occurred. So in review, the PCR, or the polymerase chain reaction, is just a process that we use to amplify DNA, and it relies on a series of reactants to produce DNA of particular fragments. And obviously it's driven by temperature, and we use a thermocycle to help this process occur. So we're also going to look at a couple of interactives as well that might show this process in real time. So I hope this video helped. Thanks.